Hello, everyone, and welcome to the Precision Medicine virtual event. My name is Ra'ed Samara. I'm a Global Product Manager at QIAGEN, and I will be presenting on Beyond the Score, Tools for Pan-Biomarker Analysis and TMB Estimation, enabling deeper understanding of somatic alterations that contribute to cancer initiation, progression, and recurrence. Please feel free to submit questions during my presentation, and I will follow up with you via email. Just as a disclaimer, the QIAseq targeted DNA panels are intended for molecular biology applications only. These products are not intended for the diagnosis, prevention, or treatment of a disease. And what I would like to go through in the first part of this presentation is to go through the chemistry aspect of a pan-cancer, pan-biomarker panel that we have developed that can be used also for TMB estimation. And then my colleague Beate will go over the bioinformatics piece. And just to start off with some background about immuno-oncology and immunotherapies, anyone who has done any work with cancer understands the hallmarks of cancer. One of the hallmarks being avoiding immune destruction. And we know that immuno-oncology approaches seek to modulate the immune system to promote anti-tumor activity and counteract this hallmark. However, for immuno-oncology approaches to work very effectively, they need to incorporate an understanding of the mechanisms of tumor escape within the tumor microenvironment. So I guess the first question that we always want to answer is what is a tumor microenvironment? And basically, the tumor microenvironment is a living organism. It is an environment that consists of many different types of cells, such as tumor cells, tumor-infiltrating lymphocytes, or TILs for short. And we know that TILs have the capability to kill tumor cells. They get primed in lymph nodes. They have the capability to infiltrate tumors, recognize these tumor cells, and become and remain activated within this tumor microenvironment. However, we know that the cells in this tumor microenvironment are in constant interactions with each other, and they can sculpt each other's activities and capabilities. And we know now from several different publications that tumor cells have devised mechanisms to avoid immune destruction. This is one of the hallmarks of cancer, even in the presence of activated TILs. So basically, an understanding of immune evasion within the tumor microenvironment is going to be key to advancing immuno-oncology research. And if we look at some examples of how tumor cells do this, we know that the T cells can recognize their targets by binding to the T cell receptor, by binding of the T cell receptor to antigens associated with MHC class 1 molecules that are presented on surface of all nucleated cells. So basically, this type of binding of a tumor cell to T cells through these receptors will lead to the activation of T cells. And this is represented on the right-hand side of the slide, where you see a tumor cell shown in yellow and the T cell shown in blue. The interaction now between these two different types of cells through the T cell receptor will lead to the activation of T cells. However, we know that immune checkpoints such as PD-1 have been devised by T cells to control their activation. T cells do not want to remain in a constant activated state, so they've devised these mechanisms of immune checkpoints such as PD-1 to control their activation. But we also know that tumor cells express PD-L1, which is a ligand that binds to PD-1. And the binding of PD-L1 to PD-1 actually inhibits T cell function. And this is shown on this part of the presentation, whereby you see how PD-L1, the binding of PD-L1 to PD-1 can actually lead to the inhibition of T cells. And the immunotherapies that have been devised so far in the form of anti-PD-L1 antibodies or anti-CTLA-4 antibodies, they actually they block the interaction of PD-1 to PD-L1. And by blocking the interaction of PD-L1 to PD-1, you now have a mechanism by which you can keep T cells in an active format. And a number of immune checkpoints operate to control the immune system. The PD-L1, PD-1 axis is only one of these forms. And this is actually shown on the next slide. There have been different types of immune checkpoints discovered that actually modulate the immune system. As you see on the lower part of the slide in green, these are the sort of the activation pathways that lead to the activation of T cells, but on the top part of the slide in red, you see these immune checkpoints. And again, PD-L1 and PD-1 interaction is only one of these immune checkpoints. The other heavily investigated checkpoint is CTLA-4, and by the binding of B712 to CTLA-4, this is another checkpoint sort of mechanism that has been devised to inhibit T cell activation. And there are a lot of other immune checkpoints, but for the sake of this presentation, I'm using only PD-L1, the interactions between PD-L1 and PD-1 as an example of this immune checkpoint. And again, immuno-oncology research has been successfully translated to immunotherapies in the clinic simply because we understand how the receptor, let's say PD-1 and the ligand PD-L1 interact and lead to the inactivation of T cells. Now, if you look at the different types of immunotherapies that have been approved so far, I'm listing 4 of them over here with their generic name and the brand name of Yervoy, Opdivo, Keytruda, and Tecentriq, along with their targets. And you see that the majority of them, actually 2 out of 4, target PD-1, Tecentriq targets PD-L1, and Yervoy targets CTLA-4. Along with their approval dates as listed on this slide. And again, keep in mind the idea behind these immuno-oncology therapies is to prevent the interaction between the ligand, such as PD-L1, to its receptor that is expressed on T cells, which is, again, in this case, PD-1, to keep these T cells active. And these different drugs have been approved for different types of diseases, such as metastatic melanoma, non-small cell lung cancer, and bladder cancer. And so immunotherapies, again, in the form of checkpoint inhibitors, have been shown to be very effective in treating a subset of these different diseases, again, such as melanoma, non-small cell lung cancer, and bladder cancer. And a lot of questions have come up as to why these types of immunotherapies are effective in only a subset of the different cancers that we see. And what I will go in the next part of this presentation is to kind of show you why these have been effective in these types of cancers and not others. But before I get into this, we now know that immunotherapies are basically a pillar of cancer treatments. Traditionally, cancer has been treated with chemotherapy, radiotherapy, and surgery. But now immunotherapy is one of those pillars of cancer treatment, and they come in different flavors. I've only spoken about checkpoint inhibitors like Yervoy that blocks the interaction between CTLA-4 and its ligands, but there are other types of immunotherapy such as cancer vaccines and CAR T cells, but I won't go into those in this presentation. So I mentioned that there are specific cancers that actually respond to these immunotherapies and immuno-oncology drugs. And again, there have been intensive research to try to answer the question of why. And this is shown actually on this slide here. You do see that, let's say, for instance, for colorectal cancer, colorectal cancer specimens actually express PD-L1, as you see on this slide on the far right hand side of this table. 53% of colorectal cancer samples actually express PD-L1. And so if they express PD-L1, generally speaking, or logically speaking, they should be responsive to the inhibition of PD-L1 and its interaction with PD-1. However, these have not translated to good responses in colorectal cancer, but you do see good responses for other cancers such as melanoma, non-small cell lung cancer, ovarian cancer, and what have you. And again, there have been a lot of efforts to try to answer the question, why not? Why do you not see good responses for colorectal cancer, for instance, when these samples actually express PD-L1? And to try to summarize everything that has been done so far, the reason why is because examining one biomarker or mechanism of action is not sufficient to unlock these secrets of immuno-oncology. And one needs to take an omics approach in order to understand why these tumors are responding, or some tumors are responding, some tumors are not. And if you try to look at this omics approach for immuno-oncology, there are several elements that can be tied into what is called an immunogram, which is basically all the different elements that can be looked at in order to understand responses to immunotherapies. One of them is tumor mutational burden, or TMB, and MSI status. One can look at lymphocyte counts. One can look at the immune repertoire profiles that exist within the tumor microenvironment or so. One can take a look at gene expressions in terms of effectors and inhibitors, soluble inhibitors, tumor metabolism, MHC expression, and neoantigens, as well as microbial profiles. These are all elements that can be, again, tied all together as in a cancer immunogram. And we know that NGS is the ideal technology for an immuno-oncology omics approach, because you could use the same technology to look at a lot of these elements. So we know that we could use NGS to assess TMB status, to assess MSI status, to look at T-cell receptors and understand the immune repertoires. We can use NGS to look at gene expression levels of different effectors and inhibitors, to examine neoantigens and their association with MHC expression, as well as microbial profiles. So it is that ideal technology for this immuno-oncology omics approach. And one of the elements that I will focus on today is, of course, the TMB mainly. MSI won't be part of this presentation, but the focus is going to be on TMB and how you could use a large panel that has been developed to be pan-cancer, pan-biomarker, to also assess tumor mutational burden status. So, but before I get into the solution, just a quick definition of what TMB is. And we all know that TMB is just basically the mutation load in tumors. We understand and know that different types of tumors express a different number of mutations per megabase. So you see on the far right-hand side of this graph, you see melanoma, lung, and bladder, and these are the tumors that express the highest number of mutations per megabase sequence. But you also see on the far left-hand side of this graph, you see those tumors that do not express a large number of mutations. And so if you want to get to a pure definition of TMB, it's just basically out of 1 million bases that you sequence, how many mutations do you see? Now, in order to address the sort of the issue of trying to answer what is the mutational tumor burden profile of a specific sample, we've devised and have developed what is called the QIAGEN TMB panel, or the QIAGEN tumor mutation burden panel, in order to assess the TMB status of different samples. And the workflow of such a panel is shown here at a very high level. It starts off with DNA that has been extracted from biological samples, and the workflow tolerates as little as 10 nanograms of DNA. This is followed by 4 main steps: fragmentation using a single enzymatic reaction to basically fragment the DNA into smaller pieces and prepare them for library construction. The second step is library construction and unique molecular index assignment. The workflow incorporates UMIs, or unique molecular indices, or identifiers into the original DNA molecules so that we can track them with our bioinformatics solutions. And then this is followed by target enrichment using a single primer extension approach, where we enrich for 486 different genes by targeting their exonic regions. All of these primers that are required for the enrichment have been pooled into an individual pool of primers. And then this is followed by library amplification, where you can multiplex up to 384 samples per sequencing run. The entire workflow takes about a day to go from extracted DNA samples all the way to libraries that can be quantified and sequenced. Now, the solution, or the approach that we've built for TMB analysis and pan-biomarker analysis in different types of cancers is a sample to insight approach, in the sense that we've taken not only the chemistry but also the necessary bioinformatics tools to allow you to analyze all of that data coming out of the panel, and we package them into one solution. So if you look at the elements that are required for TMB analysis, for instance, you need the TMB panel itself, which contains the proprietary PCR chemistry to enrich even GC-rich regions, and primers that are based on single primer extension approach for enhanced uniformity. The panel box is sequencer agnostic, in the sense that the same chemistry can be applied to different types of sequencers, like the Illumina sequencers or Iron Torrent sequencers. The sequencer specificity comes in at the index box level. This is another box that is required for the workflow, and it contains the molecularly barcoded library adapters that incorporate unique molecular indices. And then, of course, you have the analysis pipelines. These are barcode-aware data analysis pipelines that analyze the data not only on raw reads, but also on unique molecular indices. And in addition to the secondary analysis pipelines, there are interpretation pipelines as well, and solutions to allow you to basically make sense of all of the different variants that have been detected. Just to kind of elaborate a little bit on the two unique elements that we built into the chemistry side of this equation, namely the single primer extension approach and the unique molecular indices. And what I'll do in the next maybe 3 or 4 slides is to give you sort of a sense of what single primer extension is all about and what UMIs are all about and how they are helpful for such an application. So for single primer extension based enrichment, what I want to do here is to just compare it to the traditional amplicon-based approach that uses 2 primers to define a specific region. So if you think about how we traditionally do and perform amplicon-based sequencing is to define the regions of interest using 2 region-specific primers. So just as an example, I'm using exon 21 of EGFR, and if you're using the 2-primer amplicon design approach, you would need to define the exon 21 of EGFR using 2 primers that are specific to that exon. Now, while this has been very effective, let's say for smaller targets like SNV analysis or hotspot analysis, it has a lot of limitations. Some of the limitations are listed over here. You have an increased risk of primer dimers and dropouts, just because of the large number of primers that will be required, which will lead to also lower uniformities. And then multiple primer pools are going to be required in order to split those primers into different pools to maintain a specific, let's say, library fragment size. And it also leads to lower library complexity because you are defining the start and stop position of your amplicons. And now increasing the panel content might require a complete, primer redesign of the existing primer. So let's say you're using a panel of 400 genes today, and then you want to increase the content by adding another 5 genes. The additional primers that are required to cover these additional 5 genes, they will have to be basically incorporated into your original panel, and now the primers of the original panel might have to be redesigned. So in order to overcome these kinds of challenges, we've incorporated a single primer extension approach for amplicon design into the QIAseq panels. And the way single primer extension approaches work is very similar to the 2-primer amplicon design. The biggest difference is that your regions of interest are not defined by 2 primers anymore. The region of interest is defined by one region-specific primer. The other primer that is going to be required for enrichment is basically a universal primer that binds to sequences introduced in the library adapters. By doing so, we are reducing the number of primers that are required for enrichment, and that allows us to reduce the risk of primer dimers and dropouts. It allows us to achieve a higher degree of uniformity, and now only a single pool of primers will be required for enrichment, regardless of how large that region is. This also leads to a higher library complexity because only the start position is defined by that region-specific primer. The end position is completely random, and it depends on the library adapters. And now increasing the panel content does not require primer redesign because you've eliminated or reduced the risk of primer dimers and dropouts. So single primer extension is just basically a way for us to maintain the benefits of amplicon sequencing, but also overcome the challenges that we see with current approaches of amplicon sequencing. And just to kind of give you a sense of what unique molecular indices are all about and how they are beneficial for data analysis, I'm going to show you 2 examples of how we could use unique molecular indices for accurate quantification, and then on the next slide it would be for accurate variant calling. So consider the example shown, or the box shown on the top part of the slide, where you are trying to quantify, let's say, transcripts mapping back to exon 21 of EGFR as a surrogate, for EGFR expression. With conventional targeted DNA or RNA sequencing approaches that analyze data based on non-unique reads, if you have, let's say, 5 different reads mapping back to exon 21 of EGFR, it is very difficult to basically differentiate between two scenarios of having 5 unique DNA or RNA molecules or quintuplets of the same DNA or RNA molecule. Basically, you're looking at PCR duplicates. So in the top part of the slide, you basically don't have a way, you cannot really differentiate between these two different scenarios. However, if you incorporate unique molecular indices before any amplification takes place, you can now actually differentiate between these two questions. Because if you, as you see on the bottom left of the slide, if you have 5 different UMIs attached to these 5 different transcripts, you know you've started off with 5 unique transcripts, and what you're looking at are actually 5 unique transcripts mapping back to exon 21 of EGFR. But if you have the same unique molecular index across these 5 different reads, then you know you're looking only at 1 unique molecule that was just amplified over and over. So you're looking at PCR duplicates. So what this allows you to do now is to achieve a higher level of quantification for these transcripts by incorporating unique molecular indices. And UMIs are also important for accurate variant calling, and I'll use the sort of a very similar example where we're looking at mutations across exon 21 of EGFR. And if you only see, let's say, 1 mutation as denoted by this red asterisk, if you see this one mutation in 5 different reads that map back to exon 21 of EGFR, you don't have the necessary sort of statistical power to differentiate between a true low frequency mutation, let's say, that is actually part of exon 21 of EGFR in your original biological sample, or are you looking at a PCR or sequencing error, basically a false positive in your data. So with UMIs that have been incorporated, again, before any amplification takes place, you can actually now tell the difference between a true low frequency mutation and a PCR or sequencing error by examining, again, these unique molecular indices across all of those reads that map back to that specific region. So on the lower left-hand side of the slide, you see where we're looking at a false variant because that specific variant that we're looking at is actually part of 5 different reads that map back to exon 21 of EGFR that contain the same unique molecular index. If it was a true mutation, as you see on the right-hand side of that box, if it was a true low-frequency mutation, you should be able to see that mutation in all of the reads that carry the same unique molecular index because they're coming, from the same original molecule. So by incorporating unique molecular indices and analyzing data based on unique molecular indices, you now have a way to differentiate between what is a true low frequency mutation, as you see again on the right hand side of the box, the lower box, or a PCR error, as you see on the left hand side of that box. So just looking at the content of the QIAseq TMB panel. The QIAseq TMB panel covers exonic regions of 486 genes, and the genes, have been, they belong to different classes of biological processes, such as oncogenes, tumor suppressor genes, cell cycle genes, epigenetic genes, and what have you. We've covered a wide range of biological processes with this content, and the content itself is shown on this slide, but I won't go through every single gene. It is here for your reference. Looking at some specifications, as I mentioned, it covers 486 genes with more than 19,000 primers, all in an individual pool of primers. The number of target bases is about 1.3 million. The types of variants that can be called are basically TMB scores, SNVs, indels, and copy number variants. So you can actually, again, you could use this type of content and panel not just for TMB analysis, but also to look at different types of biomarkers. Specificity and uniformity are listed here at 95.5%. So 95.5% of the reads are specific and they map back to the regions of interest. Uniformity at around 20% of the mean is extremely high at 97.8%. And the recommended DNA input that we would recommend for TMB calling is 40 nanograms, even though the workflow can tolerate down to 10 nanograms of input. But if you're interested in calling TMB scores specifically, we would recommend 40 nanograms. It utilizes single primer extension for enrichment and incorporates unique molecular indices. And you can multiplex up to 384 samples on an Illumina instrument or up to 96 samples using an ion torrent instrument. Again, the total workflow time is 8 to 9 hours. Of course, this depends on the number of samples being processed, but you can go from extracted DNA to libraries that can be quantified and sequenced in 8 to 9 hours. And the variant allele frequency that you could call is down to 0.5%. And just to give you a sense of actual data coming out of this panel, specifically looking at TMB profiling or TMB status calling, we've done some experiments internally as well as with some of our collaborators to look at how do the scores coming out of this panel, how do they correlate to scores coming out of whole exome sequencing approaches, because that has been the sort of the gold standard for TMB calling. And if you look at the correlation between TMB scores out of the TMB panel, as you see on the y-axis, and TMB scores from whole exome sequencing projects, as you can see on the x-axis, you do see a very nice correlation between the two approaches. So we know that calling TMB scores from this QIAGEN panel is going to give you an extremely high degree of correlation with TMB scores called from exome sequencing approaches, but of course at a more cost-effective fashion, in a more cost-effective fashion. And not only do we compare and correlate the scores coming out of a panel with whole exome sequencing approaches, but we've also compared them to scores coming out of Foundation 1 panels, and we do see a very nice degree of correlation between the scores generated from the QIAGEN panel and the informatics solutions with Foundation 1 scores. And also just to look at the variation between site to site, we've run the same samples across 2 different sites, and when we look at the different types of TMB scores generated, we see a high degree of correlation between the first site and the second site. And so with this, I will end my part and I will turn it on to Beate.
Thank you so much, Raid. So we're going to shift here a little bit of focus on the tertiary variant analysis, which is basically the last step in the NGS sample to insight workflow and refers to the interpretation and reporting of variants. And I'm going to dive deeper into QIAGEN Clinical Insight Intepret, the tertiary analysis solution. This is the disclaimer. QIAGEN Clinical Insight Intepret is also available as clinical decision support software. So QIAGEN Intepret enables labs to deliver evidence-based insights for any indication across rare hereditary diseases, hereditary cancers, oncology, and hemato-oncology. So today we're going to focus on how QIAGEN Intepret provides biological relevant insights into variants detected from comprehensive cancer panels. So what is QIAGEN Clinical Insight Intepret? So this is at a high level an overview of the various features of QIAGEN Intepret. QIAGEN Intepret assists in the classification and reporting of alterations and is based on a comprehensive content knowledge base which allows QCI to display the alterations biology, reported clinical cases, prognostic outcomes, treatments, and trials for the particular alteration of interest in context of the user-entered disease phenotype. Therefore, the QCI platform allows you to answer important questions. What are the biological characteristics of these alterations? Is it clinically relevant, and is it actionable? QCI Intepret computes the biological relevance and actionability according to the ACMG and AMP guidelines for alteration classification, and because this removes the need for time-consuming manual research, QCI provides labs with a scalable automated interpretation solution. The QCI Intepret platform provides the user the option to customize the final report, and it can be configured towards the lab's specific needs, which may include lab-specific reporting policies, test-specific variant pre-filtering, or the integration with other existing laboratory pipelines. All of that helps the lab to automate, standardize, and scale the NGS sequencing solution. So as panels are becoming increasingly larger, customers are challenged by the increase in detected mutations, interpretation of more biomarkers, and an increase in novel variants. Customers are commonly receiving VCS from the secondary pipeline that may contain thousands of variants. And so therefore, labs using comprehensive cancer panels are challenged to prioritize and interpret relevant, actionable mutations while ensuring a fast turnaround time. So QCI can be customized via test-specific configurations, which enables consistent and scalable prioritization of clinically relevant alterations. And this can be accomplished by a customizable variant pre-filter, and I show here in the slide a few of those options that customers can select to narrow down clinically relevant alterations. Customizable preferred transcripts, customizable reporting policies, which allows the automatic reporting of alterations, treatments, and trials based on the laboratory's reporting policies, and a customizable report. So this enables an automatic prioritization of relevant variants at scale. So what situation in general are we facing when interpreting the somatic alteration? So you can see here that many resources are necessary for gathering information to understand the biological relevance of an alteration. And that means we have to sift through public or proprietary databases to see if this variant has been seen before in my disease context. And I just highlight here a few handful resources. So further, we have to dig into published literature that describes the function of this mutation, its role in tumorigenesis, and association with the disease, and potential use of variant prediction tools to predict the deleteriousness of the mutation. So then we also have to understand the clinical relevance of an alteration. So is the alteration relevant for therapeutic, prognostic, or diagnostic actionability? And that means we have to review the indications on drug labels, review constantly changing guideline recommendations, open clinical trials, and dig into the primary articles that provide insights into the actionability of an alteration. So there are challenges that we need to overcome. And I highlight a few of those. So the first one, efficiency. So this is a manual curation process that is laborious and time-consuming. The second challenge is the accuracy. As with a manual process, it can be error-prone, and especially if no standardized curation protocols and semantic consistency is used. Then the scalability. The number of articles that describe an alteration in the context of a disease, outcome, or therapeutic response has increased fourfold during the last 10 years. And a lab would basically need to hire an army of scientists to keep the variant characteristics up to date, which leads to the challenge of being comprehensive in content at a reasonable amount of cost. The QIAGEN has simplified this process by putting all of this information in one centralized location known as the QIAGEN Knowledgebase. So the QIAGEN Knowledgebase holds more than 21 million findings. What that means is that this is information that can be alteration disease associations or alteration drug response associations, and this is stemming from more than 30,000 publications. To ensure an up-to-date knowledge base, the content is updated mostly on a weekly basis, which allows us to cope with increasing number of literature sources and changing information from labels, trials, and guidelines. The amount of external databases that customers might need to review when interpreting a variant is tremendous. The QIAGEN Knowledgebase incorporates 31 external, publicly or proprietary databases, and 25 of those are used in QCI Interpret. So, with respect to the therapeutic actionability, as of today, we have more than 13,000 manually curated alteration type drug response disease associations across a variety of indications that support the therapeutic actionability in QCI Interpret. I also want to point out that the Knowledgebase provides worldwide open and recruiting clinical trials, where biomarker-required trials provide access to investigational drugs across more than 410 genes in 530 indications. To assess the prognostic relevance of an alteration, which is of particular interest for rather hematologic malignancies, the QIAGEN Knowledgebase covers content on a variety of different cancer types. So, what information specifically fuels QCI Interpret, and how is it possible that QCI Interpret can match the evidence from the literature to compute the alteration classification in context of a specific disease? And this is accomplished through a unique knowledge-based model. So what you see here is that expert curators, which are Ph.D. and MD levels, read the scientific literature, including figures, tables, supplementary information, and add each alteration, clinical effect, and drug disease association into a structured format into the knowledge base. And then we use natural language processing to put the pieces of information from the knowledge base together into a sentence which returns the essence of an experiment. And examples are, for example, functional studies or clinical trial results from a paper. So therefore, unstructured data is consistently structured with semantic consistency in the ontology, and this is done across the different sources. So we can then apply rule-based algorithms on the structured data in the knowledge base, automatically trigger pathogenicity criteria such as the PS3 or BS3 rule, or actionability tiers such as Tier 1, Tier 1A, Tier 1B, or Tier 2D. So QCI Interpret is compliant with the guidelines for interpreting alterations. The underlying guidelines to assess the biological relevance are based on the ACMG guidelines, and QCI Interpret provides the actionability classification based on the ASCO/CUP guidelines. Because assessing the pathogenicity and actionability of an alteration is time-consuming manual research, QCI is capable for providing you with the QCI computed classification of this alteration. So now, how is the information from the knowledge base displayed in QCI Interpret? So due to the limited time, I wanted to highlight here a few key features that provide the actionable or biological insights into the alteration detected in solid tumors. So what you can see on this slide is that this is a variant list of an NGS result from a stage 4 lung cancer sample. So each tile represents an alteration detected in the tumor. And the different colors, such as red or green, display the QCI computed pathogenicity of an alteration. And the letters, such as 1A or 2C, indicate the QCI computed actionability classification. So if the panel was designed for determining the TMB score or MSI status, the user will be able to review the data in the header of the test. So this view basically provides a broad overview of the patient's molecular profile and allows various filtering to prioritize the alteration that would need a deeper review. So if you would click on a tile, it would allow the user to rapidly assess the biological relevance of the alteration. So within each variant details section, QCI provides front and center an assessment section, which displays the actionability criteria and pathogenicity criteria that are automatically triggered by QC Intepret. The user is able to immediately review the description of the ACMG criteria, as well as review the underlying scientific evidence that supports the criteria. So one thing to note is that the user has still full control of adjusting the pathogenicity criteria by removing or adding a criterion, or adjusting the strength of the criteria and adding any notes why this was done. So that provides the user full control over determining the final pathogenicity assessment. So in this example, the functional consequences of an alteration can be easily assessed by clicking on the info icon to gain a snapshot if this alteration has been characterized in the literature. After reviewing the underlying evidence of the PS3 rule, which refers to established in vitro or in vivo functional assays, the user can review the underlying publications, but also look at the location of the alteration, if it's falling into an important functional domain or in an exon relative to the whole gene. So for those alterations where the function of the alteration is less clear, the association of a specific mutation in a disease of interest might be another way to shed more light into the role of the mutation. In this case, the user has the ability to review the alteration association within the context of the disease in the assessment section, as well as in more details in the reported clinical case section. So now focusing on the actionability tiers. All tiers that are applicable will be shown in the assessment section. For each tier, the underlying clinical evidence can be viewed and provides transparency in the sources that support each tier. And for the therapeutic actionability, the user can go and deep dive into the treatment section, which lists treatments and associated level of evidence in the context of the specific alteration and the tumor type. This section allows you also to choose the associated treatments and trials to be added to the final report. Just to note, the user has, again, full flexibility to filter down clinical trials according to the specificity, location, and phase, and then select those for inclusion on the final report. After reviewing the information in the varying details page, the user is able to set the final assessment of the alteration. In this section, the user has the ability to report out pathogenicity or actionability for each alteration. Before generating a final report, the review and report page provides you with a summary of the information marked as reportable during the assessment process, which provides features to complete the report building process, manage and review process, and generate the report. So for alterations that are marked as reportable, QCI can generate an interpretive comment, which contains information about the gene, alteration function, and the therapeutic relevance, which saves the user time-consuming manual work. The user is also able to review the reported drugs and review QCI-generated drug comments, which can be edited as well. So then we have a change state button that allows you to manage the review process on the test. And then when the report is ready for the sign out process, you can click on the sign out button at the top of the page. So last but not least, before signing out, you might want to review the final report. So this is a fully customizable final report, and that basically shows relevant variant information, including the ACMG pathogenicity and AMP actionability, relevant drugs, and trials. So I also want to highlight here that QCI Interpret will provide front and center on the final report the TMB score and the MSI status if those were determined during the secondary analysis. QCI Interpret results have been validated by various laboratories, which compared the QCI computer classification to manual expert review. And I wanted to draw your attention to the right side of the slide. In this study, our lab highlighted the time-saving aspect when analyzing rare variants, and they estimated across 50 patient samples time savings of up to 78% when using QCI Interpret compared to expert clinical genetics creation in large panels. So to summarize the key values of QCI Interpret, any lab can immediately adopt the ACMG or AMP guidelines for interpretation and reporting. And QCI computes those guidelines for somatic alteration classification, which provides the lab a scalable, standardized alteration interpretation and reporting solution. QCI provides for each classification full transparency to every piece of scientific evidence. It is a configurable platform that provides customizable reporting. It has flexible integration with lab systems for seamless interpretation workflow. And therefore, QCI Interpret enables fast analysis of results through scalable alteration, interpretation, and reporting. Thank you so much for your attention.